Hello. First of all, I would like to thank Vanessa for organizing this very lively conference, and I will try to get a very brief glimpse from the basic cell biologist point of view on one of the molecules which is involved in the CGG2 phenotype. First of all, the outline of the talk will be something about the protein trafficking and glycosylation, and Francois already made a very nice introduction into this part, as well as the core complex function, structure, localization, creation of the knockdown and knockout cell lines, some of the Golgi partners, as well as the mechanistic models for the Golgi, for the core complex function in the cell. So as already been said here, the eukaryotic cells, including the human cells, very complex structure. It contains a lot of different compartments, including this, including the ER ER membrane complex, as well as the Golgi complex, which is primarily involved in the glycosylation of the proteins and the lipids. But in addition to these different two compartments, a lot of other compartments include in the secretory and endocytic pathways, and communication between the different compartments is extremely essential for the survival and the functioning of the cell and survival of function of a tissue and the whole organisms. As already been pointed out here, that glycoproteins are sometimes consist about maybe half of the protein population in the cells. And the cells, about the 30% of the cells are being synthesized in the ER and are moving from ER through Golgi apparatus to the final destination in every cell type. So this movement of the proteins through the cell is very busy, and as you can see, you can imagine of the Golgi as this trophic hub has already been introduced as the the machine factory. But in fact, the thing is more complex. It's not just a finite adding of the structure, but also very crucial sorting of the molecule, different compartments in both so-called anterograde pathway from ER to the plasma membrane, as well as the retrograde pathway back to the plasma membrane to the ER. Everything's supposed to be in a very good shape and has to be very much organized. So in the cells, this communication between different compartments mostly happen in a small structure called the vesicles. And so these glycoproteins, as well as the components of the glycosylation machinery, is often be found in these vesicles. And the delivery of vesicles to the correct compartment is a crucial subject in this case. So again, it's already been introduced by Francois. The glycosylation is extremely important. It's involved about 2% of the genome. And in the Golgi, glycosylation is happening in the manner from cis to trans Golgi. To understand the Golgi glycosylation, as well as the role of the different Golgi glycosylation trafficking pattern, let me introduce one of the model which is important in cell biology field, a so-called the Golgi cisternal maturation model. So the Golgi, you can think about the Golgi membrane as outgrowth of the ER membranes. But in order to think about this, the proteins which are going from the ER membranes are packaged in small vesicles, and vesicles fuse together, creating the so-called cis-Golgi cisterna. At this point, the cargo protein, this protein which is supposed to be secreted, or channels which are supposed to be transported to the plasma membrane, or other glycosylated protein are being inside of this lumen of cis cisterna. And cis cisterna is maturing, acquiring different glycosylation enzymes, which is maturations happen from cis to trans. So if on the cis side, they have, let's say, the MAN2 and MAN1 enzymes, when on the trans side, acquiring the silylotransferase and galactotransferase. So according to this model, the cargo is always inside of this rather large cisterna, while the glycosylation enzymes and component of glycosylation machinery is constantly shuttling in the back fashion from a trans to cis fashion. So therefore, to maintain the proper concentration of the glycosylation enzyme and the other components of machinery, it's extremely important to maintain this constant trafficking of the factors from the trans to cis side. So in this, again, in this trafficking of the vesicles from one membrane to another, there's a very complex protein machinery involved in the packaging of protein into these vesicles. Then there's machinery to transfer the vesicles to the acceptor membrane, as well as a complicated machinery to dock and fuse the vesicles. I think the good analogy here will be, since majority of cells are water, so think about this thing as a small boats which has to be tugged and deliver their cargo to the next station. In this case, this tugging station, the first tugging station to tug these boats will be so-called the tethering factors, which recognize the vesicles on the larger distance and recognize the proper vesicles, sorting it out from the wrong vesicles. And this so-called the tethering factor, there are two flavors to tethering factors. And then there is another component of this thing, so-called the snare machinery, which we introduced yesterday. And that's the transmembrane protein, which is required for proper fusion of the membranes to allow the big content being delivered in the right place. So this combination of tethering factors and the snares have been found in every step of the secretory and endocytic pathway. Again, thinking about this cell, every eukaryotic cells, every human cells as a room full of two different compartments. To move from one compartment to another, you need this machinery. So the COG, or conservative organelle complex, is one of the components of this machinery. So it's localized, as you can see here, in the so-called perinuclear space, in the place which usually occupies the Golgi apparatus in the cells. Itself, it contains 8 different components which comprise the whole COG complex. And the nice thing about the nomenclature, it's all named from COG1 to COG8. These components has different molecular weights. It's a so-called peripheral membrane complex, so it's not completely attached to the membrane. It has the ability to go on and off the membranes. And it's interact with the other components of the machinery as I introduced before. And mutation in the COG complex led to the several types of congenital disorder glycosylation type 2. So again, it's a very rare disease, but because we have 8 components, 7 of them already been implicated in different CGD2 phenotypes. So to understand the nature of the defects connected to the COG complex, first we try to, again, using a cell model to downregulate different components of the COG machinery. Again, remember, the Golgi is organized from cis to trans, and it's very important to maintain the trafficking of vesicles from transcisterna back to the cisterna. So when we knock down one of the components of the COG machinery, COG3, you can see the protein is gone. What we observed is that instead of a very tight packaging of glycosylation enzyme in the perinuclear region, now a lot of this enzyme is now spread in the small vesicles all over the cell. You can see the same thing in electron micrographs, very tight Golgi, and now a very tight Golgi control, and a lot of small vesicles in containing the Golgi enzymes in the periphery of cells. Now, if you look on the, some of the marker proteins which are supposed to be glycosylated, like the lysosomal protein LAMP2 or plasma membrane protein CD44, you can see that in the electrophoresis, the mobility of this protein being changed. Now they're moving much faster, indicating that their glycosylation is being defective. So this first glimpse using the knockdown allow us to identify the glycosylation defects in the COG deficient cells. But what was missing for a long time is the uniform platform to understand the function of each component of the COG complex. We have knockdown cells, we have a patient cells, but the patient cells have different genetic background. Therefore, the idea was to use the, in this case, the HEK, the human embryonic kidney cells, as the model to make the null for each individual COG knockdown. And we used the strategy which we already introduced before, the CRISPR-Cas9 strategy, to create the null for each individual COG component. So we use the lectin stain to identify the cells that are deficient in the COG function. So lectins are the proteins which recognize different sugar moieties. In this case, some of the sugar moieties are usually not present in the plasma membrane if everything is going fine with glycosylation. So in this case, we use the GNL or Galactus navalis lectin, which recognize the terminal mannose. Mannose usually present in the glycoproteins in ER, but not in the Golgi and beyond. So in this case, if our CRISPR-Cas9 strategy is knocking out the components of the COG complex, we expect that the cells now being dysregulated for glycosylation. We expect that they now express the mannose containing glycoproteins and glycolipids on the plasma membrane. So we did the Lichtenstein, we did a single cell sorting and grow up the colonies and screen again using the Lichtenstein, Western blot, and sequencing. And as you can see here, we successfully made all 8 of the nulls component. What's interesting here, with the mutation as well as the knockdown, we still have this either small amount of the protein left in the cell, or the protein is modified, let's say clipped from a C terminus, and we still have some protein produced. Here is it's a clean null, so every subunit is being knocked down one by one. And you can see here this lectin, the GNL lectin, which is supposed to recognize only the glycoproteins at the very beginning of the glycosylation chain. And now they brightly stay in the plasma membrane of these cells. It also can be done by the so-called flow sorting, indicating the COG knockout cells are quite different from the wild-type cells. What's interesting here is that not only glycosylation of the glycoproteins being distorted, but also glycolipid being distorted as well, which can be identified here for the toxin binding, cholera-toxin binding to the cells. Cholera-toxin recognizes GM1 lipid on the plasma membrane, and now the binding of cholera-toxin is being reduced quite dramatically for the cells. So think about there is some actually extra benefit being given for the cells, so they don't glycosylate properly, but now they cannot bind cholera-toxin, so essentially they're protected with cholera-toxin. The same actually true not just for the cholera-toxin, but for the many other pathogens. So we done the other work showing that if the COG complex function is distorted, chlamydia cannot really replicate in the cells. If the COG complex function is distorted, HIV can't really replicate in the cells. So there's always a balance between the proper glycosylation of the protein for the good of the cells and the lack of glycosylation for another kind of level of protection, at least on the cell level. The cells which we created in HEK cells, they have the very distorted Golgi apparatus. Now it's very broken on the smaller vesicles and mini stacks. And what's also interesting, we found that in this completely clean null cells, very large vacuoles inside of the cells, indicating the increased phagocytosis in those cells. And that was quite unexpected, and it's something we haven't seen yet in the knockdown cells. Really, we have to go to the extremely low level of the presence of the protein to see some extra benefits. So again, we've done the knockdown cells. They have a disrupted Golgi phenotype, impaired trafficking, but they're still viable, able to freeze and thrive. So now we can introduce back the copies of the different COG components which found in the CD2 patients to understand how these mutated proteins are actually influencing the rescuing the cells, or what kind of specific defect can be found for these specific mutant cells. And now let me very briefly talk about think about mechanism, how the COG complex functions in the cell. COG complex, as I said, has 8 components, and preliminary results with the EM indicate that it has a so-called two-lobe structure. But we have 2 different structures: a tight lobe, like this dead spider, or extended lobe with a lot of arms, more close to the normal spider, or rather referred to octopus structure. So if you look in the cells and look exactly how the complex is localized and how it's distributed in the cells, what we found is that there are 2 fractions of the complex. One is the floating on the cytosol, another bound to the Golgi. The one which is bound to the Golgi is actually mostly split into the 2 subcomplexes, so-called the lobe A and lobe B. And that's the Golgi-bound COG complex, we think is a functional component of the complex. So using super-resolution microscopy which allow you to see at least 8 times better than regular light microscopy, we show that these 2 lobes of the COG complex on the Golgi, which in the previous slide I showed very nicely localized on the same compartment, now actually localized very close to each other. And in fact, the lobe B is mostly localized on a small structure, the vesicles, which are moving from one compartment to another one, indicating this kind of model is possible, that the lobe B is associated with vesicles, while lobe A is associated with the larger Golgi structure. So here's the proteome of the different factors interacting with the COG complex. And that's only one which we know at the point that probably it's much larger. How to dig into this thing? So what we've done here is that we created the cell lines where each individual COG components can be tagged with specific tag visible by different methods like Western blot and immunofluorescence. So now we can see all the components of the COG complex at once. That allows us to see through the different partners and see which of them interact with the whole complex, which of them interact with the subcomplexes. And one of the interaction was quite interesting, referring to the Francois talk, is that we've done the mass spec analysis on some of the COG partners, and here's some yet unknown transmembrane protein. One of them is the familiar to you TMM165 protein. TMM165 protein seems to be interact directly with the COG complex, and indeed it can be immunoprecipitated from the this cell from HEK cells using the antibody-specific TMM165. So we were very curious about the interaction and thought maybe that's one of the receptor of the COG on the Golgi. Unfortunately, we've done knockout of the both TMM165 and the COG, and in both cases they seems to be independent from each other. In the TMM165 knockout, the COG TMM165 is gone, but the COG subunits are still localized to the Golgi, and vice versa. In the COG knockout, the COG subunits are gone, but TMM165 is still in the Golgi. So while they're talking to each other, they are not interrelated in this respect. So it's still a mystery what this interaction means for us. So thinking about the other interaction, so we can just don't look in details. What we just show here that we can identify each of the partners of the COG complex and look which of them is interact with the subunit, which subunits interact with. So one of the interaction, very interesting interaction, was interaction with these snare molecules, the molecules responsible for the fusion of the vesicles and the membrane together. And one of this molecule in the Golgi is a molecule called GS15. It's a so-called V snare, a vesicular snare required for the fusion of the vesicles with the membrane. So the COG interact with this molecule. And that allows us to propose the following model for the COG complex function. When the vesicles approach into the membrane, the lobe B of the complex is localized to the vesicles, while lobe A is localized to the Golgi apparatus, and lobe B is interacting with this V snare. As the vesicle approaches, the two lobes interacting to each other, the interaction between the COG1, so-called COG1 subunits of the complex, and COG8 subunits of the complex. And this interaction tethers the vesicles to the Golgi membranes. That's allow you to be in a very close proximity and arrange the snare in the right orientation, allow you not only recognize the snare pairs, but also recognize the several different snare pairs. And at the end, the vesicles will fuse to the dark membrane, and COG complex will go through another cycle of the tethering. So actually, to prove the model, we made the construct which combined the COG1 and COG8 components together and made it in the background of the cells which completely lack of the COG1 and COG8. And indeed, in those cells, the lectin staining, which is very bright in the double knockout cells, is only partially rescued, indicating that the assembly of the COG complex is required for its proper function as required for the glycosylation in those cells. So at the end, I would like to summarize here is that vesicular trafficking is essential for protein glycosylation in human cells. The COG complex regulate the Golgi vesicular trafficking in this retrograde fashion. All COG subunits are essential for its function, and the COG lobe B subcomplex engage the V snare GS15 to tether the Golgi vesicles via transient interaction with lobe A. And I would like to thank all the members of my lab as well as the former members in collaborating our funding. Thank you.